Good morning, viewer. Welcome to our English version. Here's a really amazing news for you. Every year, millions suffer and thousands lost their lives to infections that were once easily treatable with the right dose of medication. Yeah, the drugs are the same. Human physiology is the same. The only difference is that microbes, such as bacteria, viruses, and fungi, yeah, they have now developed resistance to drugs designed to kill them. And this phenomenon, known as antimicrobial resistance, is rapidly rising, ringing sirens for emergency action across the globe. A new 11-year study found that, in addition to the misuse and overuse of antibiotics, long term climate warming can also increase the abundance of antibiotic resistance genes in grassland soils by nearly 24%. Higher temperatures favor the growth of Actinomycetota, a group of mostly gram-positive bacteria that naturally carry many resistance genes. So, as these bacteria become more abundant, the overall concentration of ARGs in the soil increases. The finds were published in Nature. Because it is predicted that by 2050, antimicrobial resistance could cause up to 10 million deaths each year if it is not addressed seriously. But in response, many organizations worldwide are working better understand and manage the factors that influence AMR. One widely One widely used framework is the One Health approach, which emphasizes that human health is closely linked to the health of animals and the environment they share. Our water bodies and soil around us are a major source of antibiotic resistance genes, which pathogens can acquire to survive antibiotic treatment. So far, research has not clearly shown how long term warming influences antibiotic resistance in soils. Understanding this link is important for anticipating potential risks to human health and agriculture as the climate continues to change. To better understand the association, the researchers combined a long-term outdoor field experiments with with high-tech genetic analysis. Conducted over 11 years, 2009 to 2020, the researchers set up experimental plots on a tall grass prairie where they could precisely control mimicking future climate conditions. Yeah, they used infrared heaters to keep the soil 3 degrees Celsius warmer than the natural environment, adjusted water levels to simulate droughts and extreme rainfall, and simulated harvesting and grazing by animals by cutting and removing the grass every year. Viewers, they also carried out shotgun metagenomics and also geochip, which are advanced genetic sequencing methods that read all DNA in a sample, giving a detailed view of which antibiotic resistance genes were present. Here, their experiments showed that warming makes resistance genes more mobile, allowing them to move more easily between different bacteria. And it also increased genes linked to resistance against glycopeptides and rifamycins, antibiotics that target bacteria. At the same time, resistant genes associated with plant pathogens become more common, suggesting that in a warmer world, controlling crop diseases with traditional methods may become more difficult. Soil samples were taken to the lab to grow and isolate bacteria, and then antimicrobial resistance tests were performed. The results showed that bacteria from warm plots were harder to kill, exhibiting greater resistance to 22 different antibiotics than bacteria from cooler plots. The study indicates that climate warming accelerates antimicrobial resistance in soil microbes at genetic and ecological levels with significant implication from public health and environmental sustainability. Further research is needed across diverse vegetation types and climates to build a stronger evidence base to guide agriculture and environmental management in a warming world. Behind this exclusive report.